Welcome everyone. Today we are going to talk about gustation. We are a group of 4 members including Zainab, Rachel, Karthi and myself, Jeff.
Problem statement. Due to the COVID-19 pandemic, students and teachers must adapt all physical classes to online classes in order to avoid contracting and spreading the COVID-19 virus to flatten the curve. As the universities consist of students from all around the world, they face many difficulties such as connection issues and time zone differences. Creative videos are extremely effective for gaining a better understanding of anatomical structures, resulting in students to become more engaged, which helps them achieve higher grades as they allow you to pause, rewind, or skip through the video to have class discussions or go over specific points.
Here are the objectives. In this video, you will learn about the anatomy of the tongue, describe the function and identify structure of the tongue, and to learn about the mechanism and physiology of gustation.
Hi, I'm Karthi, and I'll be presenting the anatomy of the tongue. One of the best ways to learn anatomy from the comfort of our homes is by exploring 3D models. 3D models allow us to visualize the structure and position of body parts in great detail. Let's look into the anatomy of our gustatory system. One of the most important gustatory organs is the tongue. Let me introduce you to Visible Body and Biodigital, which are online 3D anatomy websites. The structure of tongue. With 3D models, we can just isolate the tongue. And so let's begin by looking at the structure of the tongue. This is the apex, which literally means the tip. This is the body. And here's the root. We can also spot the submandibular duct here that channels saliva that is secreted from the submandibular gland. Let's look at the salivary glands.
In this model, we can see the submandibular gland, which is posterior to the sublingual glands here, you see. And the parotid glands are posterolateral to the 2 glands.
So these are the 3 glands that secrete saliva. Now let's look into the types of tongue papillae. The dorsal and the lateral surfaces of the tongue contain many papillae, which are the rough projections you feel on your tongue. There are 4 types of papillae. Let's look into it. The first type is located at the posterior of the tongue, and they arrange into a V shape. And you want to remember V for valid papillae. Here's how the close-up looks like.
This is the valent papillae, or also known as the circumvalent papillae. The second type of papillae is known as the fungiform papillae because it looks like a mushroom's head. They are located mostly at the apex and the side of the tongue. Thirdly, we have the filiform papillae. Filiform literally means it is shaped like a thread or filaments, and that's how they look like on the close-up. Lastly, we have the folded papillae, which is at the back and side of the tongue, and this is the complete structure of the tongue. And this is the lingual tonsils, which is located at the posterior of the tongue. So there are 4 papillae which are valid: fungiform, filiform, and folded papillae. And a good mnemonic to remember is vandals are fun-filled folks. So associating the structure or meaning with the labeled part of an organ and mnemonics are good ways to learn anatomy. Of those 4 papillae, only valid and fungiform papillae contain taste buds. Now, let's learn about taste buds. Taste buds are the ones to do the actual tasting, and they reside within the stratified squamous epithelial lining of the papillae. So this is the taste bud, and here's a close-up of the taste bud. This is another image that we got from Google Images, and taste bud here, it actually attaches onto the stratus squamous epithelium that I mentioned earlier, and the taste bud is made up of 3 cells. Basal cells are stem cells that divide and differentiate into supporting cells or gustatory receptor cells. Supporting cells, they nourish the gustatory receptor cells and they form the exterior wall of the taste bud. Gustatory receptor cell, they have a long protrusion, which is the microvilli, that collect the signal, so they do transduction and send signals down the sensory neuron. So let's get out a little bit and see what are the muscles involved for the tongue. The tongue consists of several muscles, intrinsic and extrinsic muscles that help it to bend, twist, and stretch so we can speak and swallow food. So here are some of the extrinsic muscles I'm showing that help the tongue to do so. Notice that all the labels here are ending with the word glossus. Glossa means tongue in Greek, so these muscles' names refer to the tongue. Anatomy becomes far more interesting when we look at it like learning a new language. And knowing the meaning of some of these words help us to remember the name in a way that makes sense instead of just blindly memorizing it. For example, there is a condition called lingua villosa nigra, which is Latin for tongue hairy black. That means the tongue becomes black from bacteria and feels hairy due to the elongation of filiform, and one of the reasons for this is poor oral hygiene. So congratulations, you just learned some Latin, and I hope your scalp is villosa and the hair stays nigra.
Physiology of taste. Taste is categorized as a chemical sensation in which various tastings containing food or beverages interact with taste receptors. Taste buds are the sensory organs that allow you to taste different flavors. There are 5 basic tastes, which are bitter, sweet, umami, salt, and sour. The basic mechanism of taste is first the dissolution in saliva, next attachment to receptors, followed by a generator potential, and then an action potential. Now let's look at the mechanism in more detail. First, let's look at the mechanism of bitter, sweet, and umami. When the tasted bitter, sweet, or umami binds to the respective GPCR, it activates the alpha subunit, which activates an enzyme called phospholipase C. This enzyme is responsible for cleaving PIP2. PIP2 is then converted into inositol trisphosphate, in short IP3. IP3 travels to the smooth endoplasmic reticulum of the gustatory cell and causes calcium ions to release. Due to calcium activation, ATP is released. When this happens, ATP acts as a neurotransmitter to affect the gustatory afferent neuron to depolarize and send an action potential to the gustatory cortex of the brain so that you can know you're tasting something bitter, sweet, or umami. On the right is a summary of what we just went through.
Now here, we will briefly explain about the physiology of salty and sour taste. Here is a small diagram talking about the process of how salty and sour taste is formed. Salty taste requires sodium ions to be present. Then, it finds the gustatory cells to allow sodium to enter into the cell. When sodium enters the cell, the polarity becomes more positive, causing voltage to change. The voltage-gated sodium channel will open and allows more sodium to diffuse in. Besides that, the calcium-gated channel will open as well, causing calcium to diffuse in. So when calcium enters the cells and binds to the vesicles, it causes things like exocytosis. The calcium also binds to snare proteins that are at the edge of the gustatory cells and activates the proteins. Then, the vesicle will bind with the gustatory cell membrane together to result in ATP molecules to leave the cell. When that happens, ATP travels to the gustatory afferent neuron, and with that, the neuron is triggered by ATP, creating action potential. Then, it will travel to the cortex and brain to let your body know that you are eating a bunch of salt. On the other hand, the sour taste is similar as well. First off, it uses potassium channel. Other than that, it uses hydrogen ions. Then, the whole process repeats the same as the salt taste, which form action potential that travels to the brain as well.
Hi, I'm Jeff. Right now, I will talk about the taste cell. The taste cell is a group of cells that are responsible for our taste perception. It has a short lifespan of 8 to 12 days. Therefore, our taste cells undergo a continual turnover where dying cells will be replaced by new cells from time to time. The constant turnover is essential to us as this event helps maintain our taste function and the structural homeostasis on the tongue. However, the regeneration efficiency of taste cells will decrease as we get older, especially after 40 years of age. Thus, older adults have a weaker sense to taste when consuming food. Taste dysfunction is a condition that our taste function is reacting abnormally or loss of taste. It is more commonly occurring in the adult population. There are several factors that lead to taste dysfunction. Firstly, virus or bacterial infection. Examples such as the COVID-19 virus that is now spreading around the world will also affect the patient's taste perception. Secondly, is due to medication, including ketofir, a drug that is used in lowering blood pressure. Next, diseases involving nerve damage such as the Bell palsy or facial muscle weakness disease might also lead to taste dysfunction. Nutrients deficiency is also one of the factors that contribute to taste dysfunction. For instance, vitamin B12 deficiencies will affect the folic acid formation, which is associated with taste sensory nerves. Other factors such as smoking and aging are also related to taste dysfunction. Here are some examples for you to understand diseases associated with taste dysfunction. Scarlet fever is a disease caused by bacterial infection. Patients who suffer scarlet fever have a whitish coating on their tongue. Besides, their tongue will also be swollen and reddish, thus some call it strawberry tongue. On the other hand, it is smoking. Cigarette contains tobacco that will harm the epithelium layer of the tongue. You can observe that the surface of the tongue becomes black. This is the result of the accumulation of dead skin cells on the tongue surface. Sjogren's syndrome is a kind of disease causing impairment of the salivary gland. Insufficient saliva production will affect the taste receptor, thus leading to taste deficit. As mentioned before, our taste cells regenerate all the time. Blister cells play an essential role in this process as it is the precursor for differentiation of the taste cells. Generally, blister cells divide the epithelium cells into daughter cells, then the daughter cells will develop into supporting cells. Lastly, these supporting cells undergo maturation, turning into gustatory receptor cells. In this video, we can conclude several points. First, the tongue is the main gustatory organ that is responsible for our taste perception. Secondly, every particular taste has its pathway to process the taste molecules. Furthermore, tools used in this video such as 3D models, mnemonics, image help us understand and learn anatomy in a more entertaining way. The thought of preferring learning anatomy physically than online shouldn't be a limitation anymore. Thank you for watching. We hope you enjoyed this video and gain knowledge about gustation.